Welcome to today's lecture. Today's topic is about the muscle contraction mechanism. Muscle. Muscle is a highly specialized tissue capable of converting chemical energy into mechanical energy primarily as a means for locomotion. In skeletal muscle, contraction is initiated by an electrical stimulus that starts in brain or spinal cord, and it is transmitted to the muscle via nerves. Nerve fibers that transmit stimuli to the skeletal muscle are called motor nerves. This is the diagrammatic representation of structure of a nerve cell. Nerves and nature of stimuli. At regular intervals along the motor nerve fiber, Schwann cells wrap around the axon of the nerve and secrete a myelin sheath that encapsulates the fiber. Between the adjacent Schwann cells, the myelin sheath comes in close contact with the axon, and these structures are referred to as the nodes of Ranvier. Myelinated nerve fibers have 30 to 40-fold greater conduction velocities than do unmyelinated fibers. Here we can see the transfer of nerve impulse from a motor neuron to a muscle fiber at the neuromuscular junction. Transmembrane potential. In resting nerve and muscle fibers, the transmembrane potential is about 90 mV. Fluids residing inside and outside of these fibers contain positive and negative ions. Generally, an excess of negative ions accumulates in the intracellular fluid and resides along the inner surface of the membrane. Whereas the reverse is true outside the cell, where an excess of positive ions assembles in opposition along the extracellular surface of the membrane. As a result, an electrical potential, charge difference, or resting membrane potential is developed across the cell membrane, positive on the outer surface and negative inside the cell. Development of these transmembrane or resting membrane potential in nerve and muscle results from 1. Active transport of ions through the membrane. 2. Selective membrane permeability to ions and small molecules. 3. The unique ionic composition of intracellular and extracellular fluids. Extracellular fluid contains high concentration of sodium and chloride ions. Intracellular fluid contains high concentrations of potassium and non-diffusible negative ions, for example, proteins and peptides. Sodium-potassium pump. Concentration gradients of sodium and potassium ion across the plasma membrane are maintained by active transport of sodium ion out of the cell and potassium ion into the cell. The system that accomplishes the active transport of sodium ion and potassium ion is commonly known as sodium-potassium pump. Energy required to pump both types of ions across the membrane against a concentration gradient is furnished by adenosine triphosphate, ATP. Concentration gradient created by transport of ions between the extracellular fluid and cytoplasm by sodium-potassium pump. Permeability of a plasma membrane is 50 to 100 times greater for the diffusion of potassium ion than sodium ion. Thus, potassium ion passes through membrane with relative ease compared to sodium ion. Non-diffusible negative ions in the intracellular fluid pass through the membrane with great difficulty. Therefore, the net flow of electrical charges across the membrane is due to the diffusion of positively charged potassium ions into extracellular spaces, leaving fewer positive charges inside the cell. As a result, oppositely charged ions line the membrane, with the positive ions on the outer surface attracting negative ions on the inner surface of the membrane, thus establishing resting membrane potential. Once the resting membrane potential is established, further flow of potassium ion out of the cell is impeded. As the membrane potential increases, the forces opposing potassium ion diffusion increases. Thus, equilibrium is established between outward diffusion of potassium ion along the concentration gradient and the force opposing outward diffusion. Neural action potential. Nerve and muscle fibers have the ability to transmit an electrical impulse, called an action potential, along their membrane surfaces. An action potential, often referred to as an electrochemical process. An action potential travels along the membrane surface of the nerve fiber and is actually a wave of reversing electrical charges as depolarization that results from chemical changes in the membrane. Mechanism of action potential. The action potential is initiated by a sudden and dramatic increase in the permeability of the membrane to sodium ion. At the same time, permeability of the membrane to potassium ion remains the same, and as a result an excess of positive charges enters the cell, reverting resting membrane potential. The entire sequence of events from initiation of an action potential to re-establishment of a resting membrane potential on the nerve fiber requires about 0.5 to 1 millisecond. Myoneural junction. The stimulus, action potential, that initiates muscle contraction is transferred from the nerve fiber to the muscle fiber at the myoneural junction or neuromuscular junction. The combined structures of the myoneural junction form a small mound on the surface of the muscle fiber, called the motor end plate. The electrical impulse of the action potential from a nerve is not strong enough to elicit a response alone. Therefore, a mechanism is required to amplify neuronal signals to the muscle fiber. When an action potential arrives at the motor end plate, it causes a chemical transmitter, acetylcholine, to be released. Acetylcholine is stored in small vesicles found in the terminal nerve branches, and the contents of a few vesicles are released with each action potential. When the sarcolemma comes in contact with acetylcholine, it becomes more permeable to sodium ion, the membrane depolarizes, and an action potential is propagated along the fiber in all directions. The effect of acetylcholine on the sarcolemma will occur only for a few milliseconds, and cholinesterase, released by motor end plate, will neutralize the stimulant. This picture explains the release of neurotransmitters at the synaptic cleft on arrival of nerve impulse. Calcium release. As an action potential transverses a muscle fiber via the T-tubules, it is ultimately responsible for calcium release from the sarcoplasmic reticulum that surrounds each myofibril. When the muscle is in a relaxed state, the concentration of calcium ions in the sarcoplasmic fluid is less than 10⁻⁷ moles a liter. The mechanism by which depolarization of the T-tubule is communicated to the sarcoplasmic reticulum involves two membrane-bound calcium channel proteins. Dihydropyridine receptor, located in T-tubules. Ryanodine receptor, located in sarcoplasmic reticulum, responds to depolarization of the T-tubule and release calcium ions into the cytoplasm of the muscle cell. After detecting calcium release, responds by calcium sequestered in the sarcoplasmic reticulum to flood into the cytoplasm of muscle cell, which initiates contraction. Contraction of skeletal muscle. Contractile proteins. Actin and myosin. Regulatory proteins. Tropomyosin and troponin. Contractile proteins. Cross bridges formed between thin and thick filament generate contractile force during contraction. Regulatory proteins assist in turning the contractile process on and off depending on the con of calcium ions in sarcoplasm. Myofibrillar proteins involved in muscle contraction. Actin, myosin, tropomyosin, troponin. For muscle to remain in relaxed state, calcium must be sequestered in the sarcoplasmic reticulum, that is, sarcoplasmic calcium concentrations less than 10⁻⁷ moles a liter. High concentration of ATP, which is complexed with magnesium, mg2+, ions, mg ATP, this complex prevents interaction of actin and myosin, cross-bridge formation. It is necessary to increase the concentration of free calcium ions to about 10⁻⁶ or 10⁻⁵ moles a liter, a 10 to 100-fold increase, to initiate contraction. In the skeletal muscle, troponin functions as the Ca ion-sensitive latch that fixes tropomyosin's location on the thin filament in the off state of contraction. Troponin T maintains a tight linkage to tropomyosin. Troponin C, calcium ion sensor of troponin complex. Troponin I inhibits actin-myosin interaction. Actin-myosin binding. When the calcium ions are released into the sarcoplasm, they preferentially bind to the troponin C subunit and induce a conformational change in the entire troponin complex. Collectively, these conformational changes result in movement of tropomyosin and troponin 1 toward the cleft of the actin filament and away from their blocking location of the filament. This exposes the myosin binding sites on actin. Myosin binding sites covered by tropomyosin, and when troponin binds to calcium ion, myosin binding sites are exposed. Formation of actomyosin. The shift of tropomyosin's position to the on state of contraction allows the myosin heads to form cross bridges between thick, that is, myosin, and thin, that is, actin filaments. These cross bridges develop a contractile force and the actin filaments in each half of the sarcomere. The protein complex formed when actin and myosin interact at the cross-bridge is called actomyosin. During the contraction, the length of actin and myosin filaments does not change. Rather, the filaments slide along each other, pulling Z discs closer to the myosin filaments, thereby decreasing the sarcomere length. The decrease in sarcomere length during contraction of skeletal muscle. The A band width is constant during all phases of muscle contraction, but the I band and H zone widths change. These widths are greatest when muscle is stretched and decrease as the muscle shortens. In severely contracted muscle, actin filaments meet, or even overlap, in the center of here band Z discs may bout on the ends of the myosin filaments. This picture shows the structure of the sarcomere during the relaxed state and when intermediate contracting stage and maximally contracted state. Energy for muscle contraction. Muscle contraction requires energy in addition to that normally consumed by resting muscle. This energy is derived from ATP, adenosine triphosphate, that is hydrolysed in the following reaction. ATP plus H2O gives ADP plus phosphate and energy. Energy contraction movement. The cross bridge is responsible for splitting ATP into ADP and PI, that is, inorganic phosphate, and for converting part of the energy released from splitting of ATP into force that slides the filaments. The rest of the energy is released as heat. Each cross bridge generates enough movement to slide the actin filament 10 nanometers. During contraction, filament sliding requires a cyclic making and breaking of cross bridges, each contributing to a small amount of contraction. Use of energy in contraction. The cycle of cross bridge contains a power stroke portion and a recovery portion. Point 1. Binding of ATP to the cross bridge at the end of the power stroke causes rapid dissociation of the cross bridge from the actin filament. 2. Recovery process. The cross bridge then changes conformation and hydrolyzes ATP to ADP and inorganic phosphate. Power stroke. The cross bridge then complexes with the actin filament and undergoes a conformation change, the power stroke. Inorganic phosphate and ADP are released from the cross bridge during the power stroke. Conformation change during the power stroke occurs in the portion of the cross bridge known as the lever arm. Binding of ATP to the cross bridge at the end of the power stroke prepares the cross bridge for next cycle. Cross bridge cycle in muscle contraction. Contraction phase. During resting state, the motor nerve action potential arrives at motor end plate. An acetylcholine-released sarcolemma and membranes depolarized, which leads to sodium ion influx into fiber. Then the action potential is transmitted via T-tubules to sarcoplasmic reticulum. Later calcium ions are released from sarcoplasmic reticulum terminal cisternae into sarcoplasm. And calcium ion binds to troponin C. The tropomyosin moves to expose the myosin binding site on actin. Leads to actin-myosin cross-bridge formation. Myosin ATPase activated and ATP gets hydrolyzed. This repeated formation and breaking of cross bridges resulting in sliding of filaments and sarcomere shortening. Relaxation of skeletal muscle. Relaxation of skeletal muscle involves re-establishment of the resting state and is measured by a decrease in muscle tension. In relaxed state, no cross bridges between actin and myosin filaments and the filaments of each sarcomere slide passively over one another. For relaxation to occur, conditions prevailing during the resting state must be re-established. That is, calcium concentration within the sarcoplasm must be reduced to 10⁻⁻⁷ moles per liter and ATP levels restored. First step in relaxation process is repolarization of the sarcolemma and returning the membrane potential to its resting value. Calcium sequestering by the sarcoplasmic reticulum is accomplished against a calcium concentration gradient. Therefore, an active pumping process is involved. This pump utilizes ATP as an energy source, that is, hydrolyzes ATP to ADP and pumps calcium back into the sarcoplasmic reticulum stored in terminal cisternae until released by a subsequent stimulus. The calcium pump is activated by increased cytosolic calcium. As free calcium concentrations in the sarcoplasm decrease, troponin senses the concentration change and allows tropomyosin and troponin I to return to the off location on the actin filament in which cross-bridge formation is inhibited. In the absence of cross bridges, tension is not generated and the stretching imposed by elastic components in the muscle causes the filaments to slide passively over one another. Relaxation phase. During the relaxation phase of muscle contraction, cholinesterase is released, leading to the breakdown of acetylcholine. This causes the sarcolemma and T-tubules to repolarize. Subsequently, the sarcoplasmic reticulum activates its calcium pump, returning calcium ion to the sarcoplasmic reticulum terminal cisternae. As a result, tropomyosin blocks the myosin binding site on actin, terminating actin-myosin cross-bridge formation. Following this, an magnesium ion complex is formed with ATP, allowing the passive sliding of filaments. Ultimately, the sarcomeres return to their resting state, completing the relaxation process. Sources of energy for muscle function. ATP is the ultimate source of energy. The contractile process requires large amount of ATP. The pumping of calcium back into the sarcoplasmic reticulum during relaxation, thus maintaining the sodium-potassium ion gradients across the sarcolemma. Regeneration of energy. In living muscle, ADP is very rapidly rephosphorylated to ATP. The immediate source of energy for ATP resynthesis is phosphocreatine. This diagram illustrates the metabolic pathways involved in energy production in muscle cells. It begins with muscle glycogen, which is broken down into glucose, 1-phosphate, and enters glycolysis. Glycolysis produces pyruvate, yielding a net gain of 3 ATP. In anaerobic conditions, pyruvate is converted into lactate, generating hydrogen ions. In aerobic conditions, pyruvate enters the tricarboxylic acid (TCA) cycle, where it is further broken down, producing carbon dioxide and high-energy hydrogen atoms. These hydrogen atoms then enter the electron transport chain in the mitochondria, where they react with oxygen supplied by the blood to form water and generate ATP. The electron transport chain is the most efficient energy-producing pathway, yielding 30 ATP along with 2 ATP from earlier processes, bringing the total to approximately 35 ATP per glucose unit. This diagram highlights both anaerobic and aerobic metabolism, showing how muscle cells generate energy under different conditions. Pathways that provide ATP synthesis by rephosphorylation or converting ADP to ATP in the living muscle continue sometimes after respiration has ceased. Under aerobic metabolism, when one molecule of glucose is split from glycogen and carried through entire sequence of reaction, the net energy yield would be 35 molecules of ATP. 3 ATP molecules are obtained from glycolysis along with 2H+, which will yield 2 ATP molecules in the electrotransport chain. At the end of glycolysis, 1 glucose molecule from glycogen will yield 2 pyruvate molecules. Each pyruvate will yield 10H+, for a total of 20H+ in TCA cycle. These 20H+ are converted, in turn, to 30 ATPs in the electron transport chain. Under limited oxygen supply, anaerobic metabolism is able to supply energy to the muscle for a short period of time. A major feature of anaerobic metabolism is that lactate accumulates in tissue. This diagram shows how cells generate energy through cellular respiration. It starts with glycolysis in the cytoplasm, where glucose is broken down into pyruvate, producing 2 ATP. The Krebs cycle then occurs in the mitochondria, releasing carbon dioxide and generating 2 ATP, while transferring high-energy electrons. Finally, the electron transport chain uses oxygen to produce 32 to 34 ATP and water, making this the most efficient energy-producing stage. Now we are at the end of today's lecture. As mentioned, today's topic is about the mechanism of muscle contraction. To summarize today's topic, muscle contraction and relaxation involve converting chemical energy into mechanical movement. Contraction begins with an electrical stimulus from the brain, spinal cord, transmitted via motor nerves, leading to depolarization and calcium release from the sarcoplasmic reticulum. Calcium binds to troponin, exposing actin binding sites for myosin, forming cross bridges that slide filaments, shortening the sarcomere. ATP powers contraction, relaxation, and ion gradient maintenance. Relaxation requires repolarization and calcium reuptake. Energy is regenerated via phosphocreatine and anaerobic metabolism, ensuring continuous muscle function. Thank you.